go to F18 fimbria, then we know, you see here the fimbria on the bacteria, we know that they're built up of proteins that form a sort of chain, and at the end of the chain, thus at the tip of the villi, you have an adenosine, a protein that can bind to the mucosa that is responsible for the adhesion, for the colonization. And this protein for F18 is called FedF. And FedF is a protein that binds to sugars on the mucosa. So you have different serotypes of F18, F18AB and AC, but that has nothing to do with the recognition of the receptor. It has to do with the major subunit. And all these fimbria, F18AB and AC, bind to the same receptor on the mucosa. So we looked in our lab in detail to the receptors that are recognized, and the receptor for the FedF fimbria, the adenosine of the E. coli, is a sugar that is located on the cell membrane. And the sugar is a blood group sugar, but it's not one blood group sugar. Different blood group sugars can interact with FedF. FedF has a groove. It has in that groove this blood group sugar fits, and this is sufficient to allow the bacteria to bind to the mucosa. What you see on this cartoon is that the fimbria come very closely to the cell membrane, and that is important to remember. So in order to colonize, F18 positive E. coli have to come very close to the surface membrane. And it's not only the sugar that interacts with the mucosa, but the fimbria also has a loop, and this loop is charged, and the charge of this loop interacts with the charge on the cell membrane, and it strengthens, increases the affinity, strengthens the interaction. So, but it indicates that there is an intimate adhesion needed.